This is the fourth and final set of notes for the IB Biology internal assessment. This section is called evaluation. You will be assessed in two areas for an overall score between 0 and 6. The first area you will be assessed in is going to be how well you explained any weaknesses or limitations in your lab. And so there's a lot of things that could cause these types of weaknesses. For example, they could relate to controlled variables that you did not control as good as you could have, or they could be due to systematic or random errors in the lab. We'll talk about this in more detail. These are the things that make you lack confidence in your conclusion. You need to discuss what impact did these limitations actually have on your result. So you need to be specific in this. The second thing you will be assessed on is how realistic are the improvements that you suggested to these limitations, and how well are they actually explained. So what could you do to modify the lab that will allow for you to be more confident in your conclusion? And are you being specific enough? And is it something that could realistically be fixed in that way in a high school lab setting? So the main focuses are the limitations to your methods, the impact that these limitations had on your results, and then realistic and relevant improvements to your methods. In this lab, you're going to want to talk about things that you know ended up limiting your data. So any weaknesses within the lab, and any sources of uncertainty in the lab, because these things also cause you to lose confidence in your result. You need to make sure you are showing the moderator that you clearly understand these issues and the impact that they had on your lab. In order to do this, you need to understand the different types of errors. Errors can be systematic, which means that in principle, if you could have foreseen these errors before the lab, you could have potentially eliminated them before you even wrote your methods. So for example, something in the procedure that led to a variable not being controlled, even though it should have been controlled. Or the equipment that you chose to use, you didn't calibrate it. And so again, these are going to be things that you could have potentially seen them before the lab started and fixed them before they ever even became a source of error. But you didn't, so now they are a source of error. So these things could be discussed, but keep in mind if you chose not to calibrate your equipment when you knew you should have, for example, the instructions in the probeware said to calibrate the equipment and you did not do that, then while you can discuss it in your errors, it is going to impact your experimental design section. Okay, you are going to get dinged in the research design section for not doing this before your lab. Sometimes the sources of error can be random. These are going to be the things that are completely out of your control. For example, when working with biological organisms, they are going to have variation within their biological tissues. That's not something that you have any control over. This is why you do multiple trials, so that you can look at like several chunks of data or averages of data sets. Or this could also be plus or minus the uncertainty on the equipment that you chose to use. As long as you chose equipment uncertainty that's within reason for the lab, then you're not going to get dinged in your methods for picking equipment that has uncertainty, but you do have to explain how that leads you to be uncertain and is a source of weakness in your investigation. What you're not going to talk about are blunders. Blunders are human errors, your own mistakes. Do not talk about these. So if you know, for example, that you made a calculation mistake in your lab, well, you're not going to like leave that calculation mistake in your lab. You're going to fix it and you're going to do correct math before that math ever gets seen by the moderator, and you're not going to talk about things like that in your lab. These are your errors. Human errors are not lab limitations or weaknesses. So you're looking for procedural errors. So I would recommend you go back through your methods and your list of controls, and you kind of compare the two and you decide, are any of these controls in my methods not controlled as much as they possibly could have been, and therefore leads to a source of error in my lab. It leads to you not being as confident as you could be in your final results. So some things potentially are like examples that are listed here. If you were doing a procedure where you were simulating wind and you chose different brands of fans to blow the wind, well, you should have chose the same kind of fan, right? The same brand, the same type of fan to simulate the same amount of wind, because otherwise you're not controlling that variable well. Different brands of cleaner used if you're trying to disinfect an area. Well, you need to use the same kind of cleaner, right? Because that would allow for the same type of cleanliness to occur. Different amounts of cleaner used when you're trying to disinfect an area. Well, if you're using 5 sprays in that area, then you need to use 5 sprays in every area. This makes the things comparable to each other. You used potato cells from 2 different species of potatoes when the organism you used was potatoes. Well, remember, organisms are subject to variation, and that variation is even greater when it's between different species than it would be from the same species. Was there something that would lead your raw data or final process data to not be as accurate as it potentially could be? For example, not enough trials, right? So a lot of times variation is going to, or raw data is going to vary a lot between the trials. So if you had done more trials, right, is there a way that that would have helped your lab be more accurate? Not coming in at the same time every single day to collect the raw data when time is relevant in your lab, right? So for example, if you're going to measure the height of a plant every day for 3 weeks, then you need to be coming in at the same time every day for 3 weeks to measure the height of the plant, because ultimately you're probably going to turn that into a rate of growth per day. And by not coming in 24 hours apart, you are changing the length of a day. And so that does affect the rate. It's going to make your rate inaccurate. And so you would need to be talking about that. Keep in mind, that's also a control issue. And things that are like a serious control issue, even though you could have missed it, and you're going to talk about it here, potentially, if the moderator thinks you should have caught that error before the lab, then they're going to ding you in research design. Remember that weaknesses are not your human errors, so don't ever talk about how you struggle to read a number on a graduated cylinder, or you couldn't count things that could have clearly been counted. If you are struggling to count things because they're small, right, and your equipment is your eyes, well, that's different. You're still not going to say you miscounted, but you're going to talk about why you were uncertain in your count. So there is a fine line there. Do not blame weaknesses in your lab on equipment by saying that it's not as accurate as it could have been. You chose that equipment, so you need to choose the most accurate equipment that's available to you. Our scales, our timers, our graduated cylinders, if you pick the right one, they are plenty accurate enough. So you cannot say you chose inaccurate equipment. The key thing to this section too is not only do you need to explain why these errors are limitations to your lab, but you also have to explain what effect they would have on your final process data. So did they have a big impact, a small impact, and overall, what impact? Did it increase the rate? Did it decrease the rate? Again, how did it impact your final process data? So here's just kind of a reminder. because X was not controlled, the resulting rate I calculated is higher than it should be because, again, you're always explaining here. Explain means because. You must state why. You must then suggest realistic improvements to the limitations. If you were to rewrite the procedure, how would you fix it, right? You already said this was a weakness. Well, how could you make it no longer a weakness, or at least mitigate it to the best of your ability? You want to be specific with this. State the exact fix, not a general fix. For example, if you need more trials because the data is not consistent, then state exactly how many more trials you would need and why this number of trials would fix this problem. Remember, it's an explain, so the why is important. If you want only one type of cleaner to be used as a disinfectant, then state exactly which one and exactly how much and exactly how long it is to be used, right? We have to justify why these are the way we're going to be fixing these specific errors. Not only do you need to be specific, but you need to be realistic. You need to think about, how could I fix it using the equipment that we have here at Newbury Park High School? Okay, not if you had access to like hundreds of thousands of dollars worth of, research level equipment, right? That's not realistic for a high school lab. So the example here is, if you want to enlarge an image to make it easier to see or count, then we have dissecting microscopes and we have light microscopes. We also have magnifying glasses. We also have cameras. You have your cell phone camera. We have computers. Okay, these are all things that you could use to enlarge the image and make it easier to see. We do not have electron microscopes. So if you suggested that this would fix your issue, this is not something that's realistic. We don't have that here at Newbury Park High School. And again, you also have to explain how this will fix the limitation or error. In the eval section, just like in the conclusion section, the key is going to be the why. Why is this an error? Why does this fix the error? You must explain. And that is actually it for the evaluation notes.